Hello everyone and welcome to our module on smooth muscle. Cardiac and skeletal muscle are considered striated muscle, and that's because when you look at these types of muscle tissues under the microscope you can see striations, which means lines or markings. On the screen is a picture of skeletal muscle under the microscope, and you can see little lines if you look closely in the muscle cells. Those are the striation of striated muscle. Smooth muscle, which we're going to talk about in this video, is different. It does not demonstrate striations under the microscope, in addition to a number of other differences from cardiac and skeletal muscle that we'll talk about in this video. Smooth muscle cells are components of organs. Unlike skeletal muscle, you do not find them attached to the bones of the body. You can find smooth muscle cells in the intestines, in the airways, in blood vessels, and they have a number of roles that are usually under the control of the autonomic nervous system, things that you can't voluntarily control. For example, smooth muscle helps to propel organ contents in the intestines. Very importantly, smooth muscle cells can change the resistance to blood flow in blood vessels. These cells contain actin and myosin like skeletal muscle cells and cardiac muscle cells. However, the actin and myosin function differently than in those striated muscle tissues. Some of the differences between smooth muscle cells and striated muscle are shown in this slide. First of all, smooth muscle cells do not depend on action potentials. In other words, they do not require membrane depolarization to contract. There are action potentials that can work on smooth muscle cells, but that's not the only way they contract, and that's a big difference from striated muscle. In addition, smooth muscle cells usually demonstrate slow, sustained contraction, and this is in contrast with other types of muscle cells, especially cardiac cells. As you may know, heart tissue demonstrates rapid, quick contraction to generate a heartbeat. That's very different from smooth muscle cells. And then finally, calcium does lead to contraction of smooth muscle cells, just like in striated muscle. But the way that calcium interacts with myosin and actin to generate contraction is different, and we're going to talk about that in the next few slides. In the video on skeletal muscle, I talk about actin and myosin in detail. Here what I'm going to focus on are the myosin light chains, because these are important regulatory elements that control smooth muscle contraction. So in the video on skeletal muscle, I talked about how actin filaments are called thin filaments and myosin filaments are called thick filaments. Well, myosin is made up of heavy and light chains, and the light chains are what are modified to control contraction in smooth muscle. For this reason, smooth muscle is sometimes called thick filament regulated, because myosin filaments are thick filaments, and those are what are modified, the myosin light chain portion of myosin filaments, in order to control contraction. In contrast, skeletal muscle is said to be thin filament regulated, because in skeletal muscle, tropomyosin is modulated in order to regulate contractions, and tropomyosin is part of the actin-thin filaments. The phosphorylation of myosin light chains is a major regulator of smooth muscle cell contraction and smooth muscle tone. The reason for this is because only phosphorylated myosin light chain will interact with actin. This is a special feature of smooth muscle cells. So by controlling the amount of phosphorylation of myosin light chains, a cell can control the amount of contraction or tone. There are two enzymes that participate in this process. Myosin light chain kinase phosphorylates myosin, and myosin light chain phosphatase dephosphorylates myosin. And here's a picture just to make this more clear. So myosin light chain, when it's phosphorylated, leads to contraction. When it is dephosphorylated, that leads to relaxation. Myosin light chain kinase will phosphorylate myosin light chain, and myosin light chain phosphatase will dephosphorylate myosin light chain. Calmodulin is a smooth muscle cell protein. It's ubiquitous, meaning there's lots of it inside of smooth muscle cells. Its role is to bind calcium, and once calcium is bound to calmodulin, that activates myosin light chain kinase, which, as we just saw on the last slide, leads to contraction. So what this means is that the big picture point is that anytime you get more calcium inside smooth muscle cells, that will lead to more contraction via this mechanism shown on the slide. I've added calcium and calmodulin to our picture here to make this more clear. When calcium and calmodulin interact with myosin light chain kinase, they make the enzyme more active. That leads to more phosphorylation of myosin light chain, and that leads to contraction. When there isn't much calcium around, then myosin light chain kinase is less active and there's less contraction. And remember, calmodulin is ubiquitous, so it's always present. So what this means is that the big picture is the more calcium you put into a smooth muscle cell, the more contraction you will get. So now that we understand that calcium leads to contraction of smooth muscle cells, let's talk about where the calcium inside of smooth muscle cells comes from. Well, first of all, smooth muscle cells have a sarcoplasmic reticulum like striated muscle cells, and the sarcoplasmic reticulum stores calcium, and that's one source of calcium for vascular smooth muscle cells. But the other important source are L-type calcium channels. These are calcium channels found on the surface of a smooth muscle cell. They allow calcium to enter the cell. Now these calcium channels also bind structures known as dihydropyridines, so for this reason, these L-type calcium channels are sometimes also called dihydropyridine receptors. If you look at the bottom left side of the screen, this chemical structure is a pyridine. If you add two hydrogens and remove one of the double bonds, you get a dihydropyridine. And structures that contain a dihydropyridine ring bind to L-type calcium channels, and for that reason, these calcium channels on smooth muscle cells are also called dihydropyridine receptors. In the cardiology section in the video on hypertension drugs, I talk about calcium channel blockers that are dihydropyridine drugs. These drugs all end in ipine, like amlodipine, felodipine, nicardipine. All of these drugs are L-type calcium channel blockers, and they're called dihydropyridine drugs because the drugs have a dihydropyridine structure within them. For example, the structure of amlodipine is shown at the bottom right side of the screen here. It contains a dihydropyridine ring, therefore it is called a dihydropyridine calcium channel blocker. And if you administer these drugs to patients, it will block the L-type calcium channels in smooth muscle cells. This will lead to less calcium into the cell and therefore relaxation. And these drugs are used to cause relaxation of smooth muscle cells in the vasculature and therefore lower blood pressure in patients with hypertension. Now let's talk about smooth muscle tone regulation. There are two major regulators of the amount of contraction or tone in smooth muscle, and we've already talked about both these things. That's the amount of calcium in the cell and the degree of phosphorylation of myosin light chain. And if you remember these two facts, it is very high yield. You can answer lots of board questions just by knowing these two elements that change smooth muscle tone. Now both of these things, the amount of calcium in the cell and the degree of phosphorylation, can be modified by various factors to alter smooth muscle tone. For example, the autonomic nervous system often modifies these factors to change smooth muscle tone, and there are local factors like local vasodilators that may modify these two things to change the tone in smooth muscle. The way that local factors and the nervous system modify the calcium in the cell or the degree of phosphorylation is by working through second messenger systems and G proteins. So let's talk about these things now. There are three major second messengers used in smooth muscle cells. Those second messengers are cyclic AMP, cyclic GMP, and inositol triphosphate or IP3. Their chemical structures are all shown on the screen here. Cyclic AMP and cyclic GMP are just modified versions of nucleotides, and IP3 has the structure on the bottom right side of the screen. These three second messengers are going to modify the levels of those controllers of smooth muscle tone we talked about before. That's how they work. So IP3 works on calcium. Inositol triphosphate triggers calcium release from the sarcoplasmic reticulum, and this leads to contraction. Cyclic AMP and cyclic GMP work on myosin light chain phosphorylation. Cyclic AMP inhibits myosin light chain kinase. This leads to relaxation. I'll show you a picture of this in a minute. And cyclic GMP activates myosin light chain phosphatase. This also leads to relaxation. But the big picture point here is that all these second messengers and all these systems work through the same two controllers of smooth muscle tone. That's calcium and the degree of phosphorylation of myosin light chain. If we go back to our picture, I've now added in these second messengers. So cyclic AMP inhibits myosin light chain kinase. That leads to less phosphorylation of myosin light chain and therefore relaxation. Cyclic GMP activates myosin light chain phosphatase, but that has the same effect. It leads to less phosphorylation of myosin light chain and therefore relaxation. IP3 is the only second messenger that leads to contraction. It triggers calcium release from the sarcoplasmic reticulum. And as we talked about earlier, anytime there's more calcium around, there'll be more calcium and calmodulin to interact with myosin light chain kinase, and that will lead to more phosphorylation of myosin light chain. Once you understand how these second messenger systems affect smooth muscle, it's then very easy to understand how various hormones and neurotransmitters modify smooth muscle tone in the body. For example, norepinephrine and epinephrine exert their effects by modifying intracellular levels of IP3 and cyclic AMP in vascular smooth muscle, and that's how they modify smooth muscle tone. Vasopressin works through IP3, adenosine works through cyclic AMP, and prostaglandins work through cyclic AMP as well. So you can read lots of basic science literature on all these systems, but the big picture point that you want to remember is the way these neurotransmitters and hormones work is by modifying second messenger systems inside of vascular smooth muscle, and that's how they modify smooth muscle tone in the body. So now we've talked about how calcium and myosin light chain phosphorylation can modify smooth muscle tone. We've also talked about the three second messengers that can affect calcium levels and degree of phosphorylation in smooth muscle cells. The last thing we need to discuss is how a local signal or a neurotransmitter activates these second messenger systems. And in order to understand that, we've got to talk about nitric oxide and G proteins, because that is the link between a local signal and a neurotransmitter and the second messenger systems found in smooth muscle cells. Nitric oxide is also called endothelial-derived relaxing factor, or EDRF, and if you remember this name, it's a very easy way to remember what nitric oxide does. It's synthesized by endothelial cells from the amino acid L-arginine. This is the structure of arginine on the screen here. Nitric oxide is just a nitrogen bound to an oxygen, and you can see that arginine has lots of nitrogens and oxygens in it that can be used to make nitric oxide. At the basal level, endothelial cells always produce some amount of nitric oxide, but there are many stimuli for increased production. These include the amount of blood flow or shear stress on the endothelial cells, also other substances like acetylcholine, bradykinin, and substance P. All of these things can cause endothelial cells to make more nitric oxide. Once nitric oxide is synthesized by endothelial cells, it diffuses into neighboring smooth muscle cells, and once inside those cells, it activates the enzyme guanylyl cyclase. What guanylyl cyclase does is convert GTP to cyclic GMP, as shown at the bottom of the screen here, and we've already talked about what cyclic GMP can do. It's one of the major second messengers. It activates myosin light chain phosphatase, as we just showed a few slides ago, and that leads to relaxation. So therefore, the net effect of nitric oxide, once made by endothelial cells, is to diffuse into smooth muscle cells and cause relaxation. Let me make a very high-yield point about nitric oxide. These substances, and in particular acetylcholine, work indirectly on smooth muscle cells through nitric oxide. They require endothelial cells for their role in relaxation of smooth muscle. That's because they trigger the endothelial cell to make nitric oxide. So if you take the endothelial cell away, then acetylcholine will no longer be able to relax vascular smooth muscle. And that's the kind of thing that comes up in board questions, this indirect mechanism of acetylcholine and other substances that work through nitric oxide. There are also a couple of drugs that work via nitric oxide. Nitroglycerin is a vasodilator. It's used in angina and heart failure. It's got the structure shown at the bottom of the screen here, and you can see that it contains lots of nitrogens and oxygens, and it's converted to nitric oxide in smooth muscle cells, and once this happens, the smooth muscle cells relax, and that's how nitric oxide works as a vasodilator. Nitroprusside is a drug used as a vasodilator in hypertensive emergency. It contains nitric oxide, so it's a direct nitric oxide donor, and it administers nitric oxide to the body and the smooth muscle cells and leads to very rapid vasodilation. That's why it's used for hypertensive emergency. The other way second messenger systems are activated is through G proteins. G proteins are found on the surface of smooth muscle cells. They can be activated by neurotransmitters like epinephrine, and they transmit signals to smooth muscle cells by working through smooth muscle cell second messenger systems. There are a number of subtypes of G proteins. There are GS proteins, GI proteins, and GQ proteins. The GS proteins lead to smooth muscle relaxation. They do this by raising the level of cyclic AMP, and as we talked about earlier, cyclic AMP is a second messenger that inhibits myosin light chain kinase and leads to relaxation. The GI proteins do the opposite. They decrease the level of cyclic AMP in smooth muscle cells. This leads to contraction. And then finally, the GQ proteins increase the amount of IP3. And as we talked about earlier, IP3 stimulates calcium release, and that leads to contraction. Here's a drawing of the GS and GI systems and how they work in vascular smooth muscle. The GS proteins get their name because they stimulate or activate adenyl cyclase. This leads to conversion of ATP into cyclic AMP, and cyclic AMP inhibits myosin light chain kinase, just like we talked about before. That leads to relaxation. I've written this at the bottom of the screen. The GS proteins stimulate adenyl cyclase. That leads to relaxation. The GI proteins get their name because they inhibit adenyl cyclase. Therefore, they have the opposite effects. They reduce the amount of cyclic AMP in the cell. So inhibition via the GI proteins of adenyl cyclase leads to contraction. It's very hard to remember all this. Just remember the big picture here, that the G proteins are part of how signals are transmitted to smooth muscle via second messenger systems. And here's a drawing of how the GQ systems work. GQ proteins activate an enzyme called phospholipase C, which synthesizes IP3, which, as we just talked about, is one of the major second messengers. It activates the sarcoplasmic reticulum to release calcium, and that leads to more contraction. So GQ proteins lead to contraction of vascular smooth muscle. I'll finish this video with this slide to remind you that G protein systems serve as a link between the autonomic nervous system and vascular smooth muscle. Shown on this table are a number of receptors used by the autonomic nervous system, and you may be familiar with these receptors like alpha receptors and beta receptors and muscarinic receptors. Also shown on the table is the G protein class that is usually linked with each receptor. So for example, you may know that stimulation of alpha-1 receptors usually leads to vasoconstriction. Well, the way alpha-1 receptors achieve this effect is by activating GQ proteins on vascular smooth muscle. And as we just talked about, activation of GQ proteins leads to constriction of vascular smooth muscle. That will increase peripheral resistance and raise the blood pressure, and that's why alpha-1 receptors are vasoconstrictors. You can also see on this slide the G protein class linked with other types of autonomic nervous system receptors. And that concludes our video on smooth muscle.